Jeff said, first of all, thank you for having us here. This has been a great sequence of talks to sit through this afternoon. And like Jeff mentioned, my colleague Sam Chen and I are going to tag team a little bit. I'm going to start out, sort of set up how we started this project, some of the background, introduce the data set we used. Essentially, I'm going to talk about the easy stuff, and then I'm going to turn it over to Dr. Chen, and he'll go more into the modeling approach that he's developed. So we started this project working with Mark Rogers from USGS, who has moved inland and is no longer working on the Great Lakes, but he came to us with this idea to look at productivity gradients in Lake Erie and look at how that affected different aspects of the fish community. So we had a master's student start out with this project. She looked into the data set that Mark Rogers brought to us to do some exploratory analysis and to start a modeling approach. She's since graduated and moved on to a PhD program, and Song has taken up continuing developing this approach. So that is what I will be telling you about. So picture's worth 1,000 words at least. So this is my background slide. This is pretty much what the issue is in Lake Erie. And hearing Steve talk about where the phosphorus comes into the lake, this is where it goes. So this picture is from 2013. One of a million images you can find on the internet showing the algal blooms that start in the Western Basin and sometimes extend into the Central Basin. So obviously productivity is an issue within Lake Erie. For those of you here who work on Lake Superior or Lake Michigan where you can actually see through water, this is perhaps culture shock for you. But so the reason that we're looking at, or that we were interested in looking at these this issue of productivity is to think about how it affects the fish community. So this is a goal taken from a 2003 document by the Lake Erie Committee that laid out a goal for the fish community in western central and the near shore of the eastern basin. And that goal was to have a cool water fish community with walleye as a key predator in those areas. And to have that be a self-sustaining community. So that's the goal. That's a management goal that's been laid out. One of the supporting objectives from that document stated that it was important to maintain certain ecosystem conditions to support that. So the idea was that having mesotrophic conditions and a certain amount of transparency in the water would be a supporting objective for reaching that management goal. The transparency goal that was listed in that document was 3 to 5 meters secchi depth. This is my measurement. Taking a secchi depth measurement in the Central Basin near Stone Lab. Looks pretty good there. I can say from experience that we don't often see 5 meter secchi depths in the Western Basin. So the idea that coupled with the picture we started out with suggests that productivity is exceeding what might be optimal for in some places for reaching the goal. So as you may know, and as the picture suggests, there's gradients of productivity across Lake Erie. So the depth as you go from the western basin to the eastern basin increases, but it's here in the western basin that you have the highest levels of nutrient input and consequently the highest levels of productivity. So that's one gradient, one big scale gradient going from west to east on these different factors. There's also inshore, offshore gradient. So obviously as you go from inshore to offshore, it gets deeper, at least in most places, but you're also having a gradient where the near shore area tends to have higher productivity, higher nutrient levels. So multiple gradients at different scales going across the system. Okay. So I'm going to just tell you a little bit about the data set that we used and some of the patterns that we were able to pull out of it. So our goal was to essentially look at whether certain different aspects of the fish community, specifically hercat species abundance, because the goal is a cool water fish community with walleye as a dominant predator, whether that's associated with productivity. So the way we started out looking at that was using a cart analysis, and I'll talk a little bit about what that involves. But first, the data. So these data were collected by USGS as part of the intensive CSMI year in Lake Erie in 2014. So Mark Rogers and other colleagues of his from the USGS station on Lake Erie visited 5 different transects going from the Maumee River, coming out of the Detroit River, and over to the edge of the Western Basin. So they sampled 5 transects. Each transect had 3 distances from shore. At each inshore offshore distance, they did replicates, and these data were collected in May, July, and September, so across the season. So 1 year of data in Lake Erie spread across both the inshore-offshore gradient and the east to west gradient. Ooh, whoa. Oh. Well, okay. I have no idea what this what we were supposed to see before you got the whole talk was the response variables that we were using, and if someone's trying to possibly regain control of my slides for the guys who took it over, the response variables that we had, we have the fish biomass, so we can look at the biomass of the entire community, and then we have individual species biomass. We focused on looking at perch and walleye. We also looked at those in terms of being a percentage of the community. So those are the response variables that we were looking at. We also had a suite of predictor variables that are the lower trophic level components. So we had nutrients like total phosphorus, total nitrogen, chlorophyll, zooplankton, benthic invertebrates, forage and forage fish. And some physical parameters also, including temperature, dissolved oxygen. Temperature and dissolved oxygen were the prime part. Is you're trying to break your response variable apart into groups using your predictor variable in a way that minimizes variability within groups of the response variable, but maximizes variability between groups. So the way I think of it, technically this is not correct, but the way I think of it is like the reverse ANOVA. So in an ANOVA, you start out with groups and you look at the you compare the variability between them. You're finding groupings that put the most separation between those groups. So you take one big data set that has a lot of variability and you break it into smaller categories that have smaller within group variance. So we were able to do that with our data set. And when we started out looking at total fish biomass. This is the most gross, measure of the fish community that we can now. Should I try to advance?
Yes.
Okay.
Oh, wait. Now try. Well.
Okay, it's there. Okay.
Hey, Chris.
Yeah.
Could you stand behind the mic too? Some people in the back are having harder.
Okay. All right.
Sorry about that.
So that was the tap dance part of the.
The mic still worked though.
Okay. So let's jump ahead to. So we did these categories. We were able to break the data set into pieces. We started out. I'm going to talk about total biomass just as the most sort of gross measure of the fish community. And the first way that we saw the data split in terms of looking at fish biomass was whether or not the sample was taken in the Maumee River plume or out of the Maumee River plume. So by separating out the Maumee River from the rest of the lake, you got a lot of separation in terms of fish biomass. So you're either in the plume or out of the plume. samples taken in the plume had much higher total fish biomass than samples that were taken out of the plume. So we just scientifically and statistically proved that the Maumee River plume is different from the rest of Lake Erie. I mean, it sounds kind of silly, but that actually, is good because we're just looking at patterns in the data, and that's showing us that we're seeing what we expect to see in this data set. So, when I say that when you're in the Maumee River plume, that means physically, those are samples that were located in that transect. We weren't looking, we weren't allowing the location of the plume to move, even though it really does. Those are just from that individual transect. So we can drill down into that biomass data a little bit more. And what we see is, the first thing we see is that these samples, the fish data were collected with bottom trawls. So you can kind of see that freshwater drum are a dominant species in that catch. And that most species follow this pattern of more biomass in the plume transect than out of the plume transect. The one exception was yellow perch, where biomass was actually higher in other locations. But most of the species, including drum, walleye, most of the other commonly caught species were high in the Maumee River plume. So then you continue to split that data set apart. So on this side, we're looking at things that are in the plume. On this side, we're looking at things that are out of the Maumee River plume. Once you're within the Maumee River plume, the next way the data split apart was whether you were in the near shore or offshore. So the near shore sites, again, as you would expect, had higher biomass than sites that were offshore. So again, seeing what we expect to see in terms of pattern in the data set. Outside of the Maumee River plume, we saw that we could split the fish biomass data apart based on total phosphorus. So once you're outside of that plume, you see an effect of productivity gradient. So in places where it was relatively more oligotrophic, there's less biomass than in more mesotrophic sites. So those were the general patterns that we saw out of the data set. And again, you can drill down into that a little bit more. And in terms of looking at the inshore-offshore gradient, most species, including both walleye and yellow perch, showed the same gradient, showed the same trend. The blue is the inshore and middle shore, red is the offshore site. So all of the species that made up that total biomass catch showed higher biomass in the more productive inshore areas. And outside of the plume, both walleye and yellow perch showed the same trend relative to productivity. So, and as did almost all species, where you see higher biomass where there are higher phosphorus levels. So that's where I'm going to stop and just turn this over to Sam, who's going to go on and talk about the modeling approach that we are using to then go on to look at trophic transfer.
Okay, so I'm going to talk about how we built the model after a look at the data, how we evaluate the model, and how we later on update the model. The first step in building a model like this is to turn the conceptual food web diagram to a model that the computer can use. And this kind of model is called a graphical model or directed acyclical diagram. And the acronym is DAG. I guess because I don't want to call my model DAG. So, and once you have this DAG, typically we use a technique called a Bayesian network model. A Bayesian network model will go into each node, total phosphorus, total nitrogen, and break that down to categorical variables. So for example, total phosphorus, I can break it down to low, medium, high. And when we look at this particular situation, we can't do that. If I break it down to low, medium, high, I might as well say it's east, central, western basin. So in order to get around with this, several years ago, developed a variation of this model called a continuous variable Bayesian network model, and that's the acronym we used there, CBN, continuous Bayesian network. And the difficulty with this type of model is, first of all, we have to identify all these variables to use in a DAG. Now, we have all these simple variables already, and once you identify the variables, you have to identify the functional form linking different variables through the DAG. And once you have the functional forms, as you can see, there are several layers of the model. You have to be able to quantify the parameters, parameterize those functions. And because there are a lot of parameters, and typically when we fit the model, we don't have enough data points to fit a model with hundreds of parameters. And this is a common problem we call the curse of dimensionality. It's the more dimension you have, the more data points you need. And there are lots of names to describe the situation in statistics. So this is a difficult problem to deal with. And the way we deal with that is a stepwise approach. First step is to isolate the diagram into component models and fit those component models, and then connect individual component models with a method we call a conditional modeling approach. And once with the model connected, we assess the model using a Monte Carlo simulation. Let the model forecast what the observed response variable, in this case, total fish biomass would be. And then because we use 1 year worth of data, the model is obviously not perfect, to the least. So we have to repeatedly update the model with any kind of data we have, and through repeated use of something called a Bayes theorem. Okay, so let me talk about the steps moving from a DAG to a CBN, a continuous loop. So this is the diagram I drew up when we published the paper in 2015. So on the left-hand side, you see a diagram connecting x1, x2 to a response variable y1, and then connecting x3 at y1 to y2. And if we want to isolate them, we have two different models in here, because in this diagram, you have two variables have arrows pointing to them. So the first model, you want to model the relationship between x1, x2, and y1. And suppose this is a linear regression model. Whoops. Anyway, suppose this is a linear regression model, then in this case, you will have three parameters to estimate: alpha 0, alpha 1, alpha 2. And a residual term. Residual term is parameterized by a residual variance. So if you look at the red circle on the right, that just breaks down the DAG into a continuous model, specifying x1, x2, plus the model coefficients predicts the mean of your y. And the mean, true mean of y, plus the error, observational error, gives you the data point y1. Now, when we fit the next model, which is the green circle, we can do the same thing, but connecting them. Once we fit the model, we can connect them. Connecting them, if you look at the place where after beta 2 is mu1, is the actual true means connected from one model to the other. The observation error is dropped out. And this is the essence of fitting a continuous Bayesian network model. Okay, back to this DAG. Look at this DAG. You have 6 nodes with arrows pointing to them. That means we have to fit 6 different models, and after fitting the 6 different models, we connect them together. So I'm going to just skip all the details, show you. The first model would be the Crawford-A model. Once you fit the Crawford-A model, connect that to DO model. Now, we're bypassing the observed Crawford-A data directly through the estimated Crawford-A to the DO, and then the whole model. Okay, believe me, this writing code for this is tedious, to the least. Fortunately, we had a brilliant master student, and her initial code was 20 pages long, and it took about 24 hours to run on her laptop. And once she graduated, I had to do this. I looked it over and I say, I need to simplify this. So I simplified the code to 2 pages, and was able to run this on my Chromebook for 20 minutes. So obviously, I'm a better programmer. All right, so then how big the model is. So how big the model is, once we fit the model, we have the model coefficients, and we can let the model predict what the fish biomass is, using only total phosphorus, total nitrogen, and temperature. And this is what the model predicted and compared to the data. So if you look at the comparison, the model prediction is right on, except we are predicting a little bit more variability. And that little bit more variability has all these connections in there. Okay, so that's pretty good. So then we changed the model a little bit, replacing the total fish biomass with yellow perch biomass, and adding another node, the prey fish, used to predict the. And that model wasn't that good, because the last node from benthos, zooplankton, and visibility, and prey fish to predict yellow perch biomass with R-squared value. If you fit the individual models, R-squared value is only 6%. So it's really bad model individually. When you connect them together, that's the weakest link. Even though we have a very bad component model, this entire model, you can still see the prediction is not too bad. It captures the actual data essence. All right, you have a complicated model, runs okay. Then what? Then what means, what's the use of this model? Well, to use this model, we want to actually use the model to evaluate some kind of a policy or management practice. What's their effect on the fisheries? Okay, one effect we tried was to model what would be the effect of nutrient reduction by, say, loading reduction by 40%. What the effect would be on fish biomass. And the way to deal with this is a multi-power simulation. Again, this time you enter the nutrient concentration at the reduced rate and see what happens in the fish biomass. And since the 40% reduction in phosphorus loading, not phosphorus concentration, and we didn't really want to add more work. I didn't want to add more work to myself to build another model linking the loading to concentration. So I just made an optimistic, so best case scenario type of situation. Say, let's say 40% reduction leads to 40% reduction of concentration. What's going to happen? All right, so that simulation gives you the top bar. And you see the on the lower left bottom shows you that box plot represents about a 30% reduction in biomass. All right, so this is the model where all the links are very strong. What would happen if I use that to predict the yellow perch? I would have a weak link at the very end. It says 5%. Well, do you believe that? I probably don't, because I know the data. When we have yellow perch biomass, lots of zeros in there, and we have to reduce the sample size by a lot. Therefore, that model is not very reliable. Okay, so you have a model, you can do different things, but at this point, the model is developed based on 1 year worth of data, and that 1 year worth of data really is a snapshot. And we fitted it for the entire lake area. What's the next step? Well, I see this model as a starting point, not as a final product. The reason being, this whole Bayesian network modeling, the structure is very, very flexible. For example, if we want to address the question Todd asked, what would be the effect of reduced nutrient loading on zebra muscle and the quadruped muscle, well, we have a DO model in here. Can we add a DO model, add a link to the muscle? We can, if we have the data. And if we have enough data to make that the component model, we can automatically add it in there. And another thing is, well, this is for the entire lake. Who cares about the entire lake? We all know Western Basin has the highest fish biomass. And you have 3 basins that are different. So, and also because this is 1 year worth of data. You say 2014 may be a good year or bad year. What happens next time when you do survey? And to address that part, I need to introduce you the concept of Bayesian statistics. In Bayesian statistics, when we have a parameter or anything that we want to estimate, we don't know exactly what that is. We always start with what we call a prior. That's the knowledge we have on that parameter before we see the data. Here's a prior represented in the probability distribution. And then you start to collect the data. The data will show you what that parameter distribution would be. And once you see those two, you apply something called a Bayes theorem, get the posterior. And now this green one is what you're supposed to believe, the model parameter. And you say, why should I believe the green one? Why should I believe the data? Well, let me ask you to think, suppose you're a Major League Baseball manager. Now, you want to steal a rookie from another team. He's supposed to be really good. You have his performance in the first, say, 12 games in the first season. And in his first 12 games, he's batting average 500. Incredible. Do you want to bet he's that good for the entire season? Obviously not, because you know your average batting average for your team is only 300. So when you want to make projection of that rookie for the entire season, you want to move his batting average towards the 300. And this is what the Bayes theorems do. Well, why do we think about that as a baseball player? Because you can think about the current model for the entire lake as a prior, the data from Western Basin as that particular rookie. Now, the Bayes theorem gives you the way to naturally combine the entire lake model with the Lake Erie, the Western Basin specific one, to become your Western Basin model. And there are papers after papers, mathematics or popular science papers to show you this is the best way of doing things. And you can do this over time. Every year, the previous year's model becomes prior, and this year's data, combine that, becomes posterior. And next year, this year's posterior becomes prior. Over time, if there's a true trend in there, it will stabilize. So you wouldn't mistake a 1 year uptick, for example, prime rate, to be the highest in 50 years. Well, why we stay at Lake Erie? We can move Lake Erie's model, if we believe the structure is right, to other lakes, other similar lakes, as long as we treat the model, current model we have, as a prior. The data from other lakes, combine the prior, you have a model for that particular lake. Okay, with that I want to add one more thing, just to say, this is the best way to create a model for stakeholders, because the starting point of this model is that diagram, the DAG. Stakeholders can input, say, what do we want to do? What do we want to use? Or what do we want to connect that to it? So we didn't think about the zebra muscle. If we have thought about that, we could add that in there. So that's an example. And with that, I want to acknowledge Jeff Tyson and Chris Vandergloot for repeated consultations in developing this DAG. And I want to say this whole process could be repeated if we have, continued project.